This is on Chapter 2, the molecules of the cell, for biology. I almost said chemistry. For biology 95. So in order to do biology, you actually have to know some basic chemistry. So I want to tell you that anything that is bold is usually one of the definitions that we ask you to know. It's on the review questions for each chapter. I would make sure that you really study these. Reading biology is usually not enough. You usually have to make like cue cards or recipe cards. You can go to the dollar store. You can make electronic ones so you have it always on your phone. But make cue cards for all of the ones, especially the ones that you find on the review sheet. So matter for sure is one. Matter is just anything that takes up space and has mass. There's 3 states of matter. There's solid, liquid, and gas. So all matter, living or non-living, is actually made up of elements. If you have done any chemistry or even a basic chemistry knowledge, you know that we have what's called a table of elements, and so that table of elements contains all the elements that we know about currently. Elements are substances that can't be broken down into simpler substances by ordinary chemical means. So, like sodium is an example of an element. Chlorine is an example of an element. Oxygen, which you usually write as O2, is an example of an element. All of these are elements, though. You'll see that they have no charge on them, so they don't have like what we would have like Na positive. This is considered an ion, so this is not an element because it has a charge. So an element would have no charge and can't be broken down into anything else, which just means that it can't be made out of anything else. It's only that. For instance, this one, which everybody knows, right, is water. That's not an element either, right, because it has not only hydrogen, but it also has oxygen in it. So that's not an element either. We consider that one a compound. 92 naturally occurring elements serve as building blocks for all the matter. Other elements have been human-made and are not biologically important. So 92 of them are naturally occurring. We just find them in the earth all by themselves, and man hasn't made them. Here you can just see that the blue ones are the ones that come from the earth's crust. These red ones are the ones that you would find in organisms. So something like ourselves, you would find oxygen, you'll find carbon, you'll find hydrogen, nitrogen, and also calcium. So just that you see what you find where. This particular acronym, so it almost looks like CHOPS, but it's actually CHNOPS, right? Because it has the N in there. This is the things that make up 95% of us. So 95% of us is made out of carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. This acronym makes it easier for us to memorize the things that we're actually made out of. Now we're going to look at atomic structure. So an atom is the smallest, so again there's one of those bold words, right? An atom is one of the smallest parts of an element, and it displays the properties of the element. It's important that you know that atoms are made out of 3 things. So atoms are made out of, number 1, protons, and I always remember that proton starts with a P. That means that they're positively charged. And the other important part about this is where they're found. So these ones are actually found in the nucleus. The other one that they're made out of is neutrons. Neutrons starts with an N, so that reminds me that not only are they uncharged, but another word for uncharged is neutral. Right? And neutral starts with N, so then I can remember them. And it's also found in the nucleus. So these 2 are in the nucleus. And then electrons is the third one. And electrons doesn't fit any of these other rules, but electrons is negatively charged. And they move around the nucleus. So we'll see in the next picture here what that looks like. So here you can see that these brown ones are the protons, and they're positively charged. These green ones are the neutrons. Remember that those ones have no charge. And then here, so this kind of area here, this is where we would find the electrons. The electrons go around the nucleus in a thing called an orbital. So they have different orbitals. The very first orbital, so this orbital here, can hold up to 2 electrons. So here's 1 electron and here's another. That's the maximum that, no matter what the element is, that it can hold in its atomic shell. This table here is just showing you a brief overview of all those things. So it shows you that protons are positive, neutrons have no charge, electrons are negative. And also where you find them, so that you find these first two in the nucleus, and again you find this one in the shell, or sometimes it's called an orbital. So shell or orbital. Now this is another way to represent chemicals, especially with carbon. Carbon is a very common one to do it this way because there's different kinds of carbon. You probably heard of carbon-13 dating. That carbon we actually write like this, so it looks like that. This is called carbon-12, but you have to know a couple things about when a symbol is written like this. First off, that C still stands for carbon, but it could be other things that we can write that way. So we can write like sodium that way and have sodium with its numbers over here too. But in this particular case, it's carbon. And again, carbon is the one that happens the most like this, that you'll happen to see like this. This thing here is what's called its atomic number. So its atomic number is like when you look on the periodic table and you find carbon, here's what it'll look like on the periodic table, right? And then it usually has the number 6 right here. That 6 is that same 6 that's here. So that is its atomic number. That thing tells us a couple things. It tells us not only the number of protons that we're going to find in that nucleus, but it actually also tells us, so we should have another one down here, but it should say that it tells us the number of electrons. Now this is not together, so like if with this carbon in particular, this means that this one has 6 protons. It also means it has 6 electrons. And I write electrons like this to show that it's negative. I write protons like this to show that it's positive. Now this number up here, often you see this one written underneath the symbol when you look at a periodic table, but it looks like this. This is called its mass number. So that mass number, or its atomic mass, those are the same thing. So these two are the same thing. What that means is that is the number of protons plus the number of neutrons. So what that means is that if I wanted to figure out how many neutrons carbon had in this particular carbon, what I do is I go, okay, 12 is the protons plus the neutrons. And if I know 6 is equal to the protons, if I subtract those two numbers, that gives me 6, right? So that 6 tells me that that is also the number of neutrons that it has. So it tells me the number of neutrons it also has. So we're going to look at some other ones though, just so we can practice some more. Some other examples of it are, so we can have this one that we just looked at, so the 12C6, we can have carbon 13, and the other one is carbon 14, so it looks like this. All of these carbons are different types of carbon. Every single one of these has the same atomic mass number though. So all of them have 6 as atomic mass number. That means that all of them have 6 protons. It also means that all of them have 6 electrons. The only thing that differs between each one of these is actually the number of neutrons that they have. So just by taking the top number, which again is the mass number, and taking this number, which is called the atomic number, away from each other, I can figure out that this one, for instance, has 6 neutrons. This one then, so 13 minus 6 is 7 neutrons. And this one, 14 minus 6, is 8 neutrons. So otherwise they're exactly the same. So this is about the periodic table. Even though each element consists of a different atom, certain chemical and physical properties actually reoccur. And we actually see this when we look at the periodic table in terms of either groups or periods. So groups are when we see the vertical columns, so things that go this way, so up and down. So you'll see that they have common characteristics, like everything in the very first row of the periodic table, for instance, when it becomes an ion, gives away 1 electron, or sorry, loses 1 electron. And then going across the rows, so going horizontally, we'll see that they have common characteristics too. They're all trying to be like this noble gas that's over here. So this is an example of a periodic table. the only ones that you really need to concentrate on knowing are sodium, oxygen, and chlorine, just because there is that one question in your review questions that's asking you to draw the atoms of two and the ion of another one. Isotopes. Isotopes are atoms of the same element. Iso actually means same in Latin, so isotopes actually mean same in Latin, with different number of neutrons. And we've already looked at this when we looked at the way that we write that. Remember that this is the atomic mass number, so this is how much a carbon weighs. This is its atomic number. Remember that, again, that this tells us the number of protons. It also tells us the number of electrons. This one here, however, tells us the protons plus the neutrons. So these are three different types of carbon. They're all still carbon because you'll see the symbol is still carbon. You also see that their atomic number hasn't changed, so it's still 6, which tells us that it's actually carbon. It's kind of almost like its name. It doesn't change. The only thing that's actually going to change here is now this number, which is the atomic mass number. To figure out the number of neutrons for each, then remember that I would just take 14 and minus 6 from it, so that would give me 8 neutrons. This one would have 7 neutrons, and this one would have 6 neutrons. All of these carbons would have, again, 6 protons because the number is 6, and 6 electrons because the atomic number is 6 again. We use the different types of carbon and radioactive substances to do a couple things in biology. One of them is that we use them to find scans for instance, in a thyroid scan. What they do is they inject you with radioactive iodine, and what will happen is it goes into the thyroid. So this is the part of your thyroid where you find it in your throat. And then this is what it looks like when it's injected with the radioactive iodine. You'll see that the areas of red are the areas that are working really hard, right? And the areas that are yellow are kind of moderate, and then blue is a little bit less but still working. Notice that there is this big area here in red that I've circled that is not showing up on the scan, right? It's like over here it's there, but here it's not there. That would indicate the presence of a tumor, so they can actually see that. The other kind of use of low-level radiation is that we use it for positron emission tomography, which is called a PET scan. What this does is you often see it on TV, but they'll show how the brain actually works. And they'll ask often questions about, like, say your name, and then they might see what areas of your brain light up when you say your name. So they're looking at studying the brain, for instance. The other uses of high levels of radiation is actually surprising, and most of the fruit that's actually in the grocery store has been radiated, and the reason that it has been radiated is because it doesn't spoil as fast then, so it actually kills the bacteria and the fungi that might be present on the fruit. Physicians also use radiation to kill cancer cells. Next, we're going to talk about electrons. So remember that electrons, again, are negatively charged, and that we find them orbiting around the nucleus, right? So they orbit around the nucleus. The positive charges of protons in the nucleus are balanced out by the negative charges of electrons. This would be in an element, for instance. So like in our carbon example, remember that carbon's atomic number is 6, right? That means that it has 6 protons, but it also has 6 electrons. And so when we add this together, we get 0 is all it's telling us. The electrons move around the nucleus in orbitals, so we've already seen that. The electrons move in energy levels, so they have more than one orbital. The first orbital or electron shell can only hold up to 2 electrons, but every shell after that can hold up to 8, and that actually has a name. It's called the octet rule. So for instance, here's hydrogen. Hydrogen is number 1 on the periodic table. So again, here is the nucleus. This is where we would find the protons and the neutrons. This is the electron in blue here, and this ring that goes around is the orbital. Next is carbon. Remember that carbon is number 6 on the periodic table. That means it should have 6 electrons. So again, here's the nucleus of it, and here are the electrons. So 2 in the first shell, because that's all the first shell can actually hold, and then the shell after that has 4 electrons in it. Again, you see hydrogen and carbon, but there's some other ones here. This one in particular is one that you have to draw for your assignment, but oxygen is one of them. Notice that oxygen has 8 electrons, so 2 in this shell, and then 6 in this shell, to add up to 8 electrons. Now we're going to look at the difference between molecules and compounds. Molecules form when 2 or more of the same elements bond together. So oxygen is an example of that. When we write oxygen, we write O2. When we write hydrogen, we write H2. When we write nitrogen, we write N2. Those things like to happen in twos. They don't like to be by themselves. Compounds form when 2 or more elements bond together. So things like water would be an example of that. Carbon dioxide would be an example of that. But that's the difference between molecules and compounds. And again, notice that the words are bolded, right? So these are words that you should probably know the definition. Ionic bonding and covalent bonding are the 2 types of bonding that we're going to look at in biology. Ionic bonding always happens between a metal and a non-metal. Sometimes we call a metal a cation, so that's just another name for it. Cation means that it's positively charged, and a non-metal is called an anion, and all that means is that it's negatively charged. So we're going to look at an example on the next page, but sodium, for instance, would be a cation here. And what you're going to see with sodium is it's going to lose 1 electron, and chlorine is the anion in this case. And what you're going to see in this case is that it's actually going to gain 1 electron. The other thing about ionic bonding that you should know is that it's transferring electrons. So it's not about sharing electrons, but it's about transferring electrons. So 1 gives up electrons, and 1 takes them. When we look at the covalent bonding, we're going to see that they actually share them. Sometimes it's 50-50 sharing, sometimes it's not, but we're going to see that they actually share the electrons. It's not about giving and taking or transferring, but ionic bonding is about transferring them. The bond between a metal and a non-metal, or between the cation and the anion, is called an ionic bond. So this picture here shows us the 2. So this is a sodium atom. So this is when it is an element. You'll notice that sodium is number 11 on the periodic table, which means it should have 11 electrons. So it has 2 in this shell, it has 8 in this shell, and then it has 1 in this shell. Now also remember that we said that they like to get that octet rule, right? So they like to have 8 in their outermost shell. So for sodium, it's actually easier for it to lose this 1 electron rather than for it to gain 7 more. So this is when sodium becomes the Na positive ion. Chlorine, in the other case, is a little bit different. Chlorine has 2, then 8, and then 7 electrons. What it likes to do is it likes to gain 1 electron because it's easier for it to gain 1 electron than to lose 1 electron, or to lose 7 electrons, sorry. So it actually gains that one. And you can even see that the sodium electron was represented with red, and here it is now orbiting around the chlorine. Notice that the shells are not connected at all. Again, because they're transferring, 1 is giving and 1 is taking. So in this case, sodium is giving up its electron and chlorine is taking that electron. So this is an example of an ionic bond. This is just what table salt looks like. So NaCl is table salt looks like once it becomes more of a. Covalent bonding results when atoms are shared. So the key word here is shared. The other thing that you should know is that it happens between 2 nonmetals. So a nonmetal and another nonmetal. Sometimes we have 50-50 sharing, so there's equal sharing. When there is equal sharing, we call this nonpolar. When the sharing is unequal, so maybe 75% of the time it might orbit around one and 25% of the time on another, that non-equal sharing is called polar. And only covalent makes polar or nonpolar bonds. So for instance, this is an example of hydrogen. Hydrogen, what you'll see is that we said, remember that this is a molecule, right? It happens in twos. With hydrogen, each one of them has 1 electron. So for them to be happy, this is actually why they go in twos. They would have to have 2 in their outermost shell. In this case, they don't have to fit the octet rule because 2 is the most that they can hold. And so if I look for this particular electron, if it came from this hydrogen, 50% of the time it'll be here, and 50% of the time it'll be orbiting around this one. Whereas in this one came from this one, we'll say. Again, 50% of the time it will be here. That is a 50, and 50% of the time it will be here. Same thing happens with oxygen. Oxygen shares a couple more electrons though. Because it shares more, it makes a double bond. We represent double bonds in chemistry and biology with 2 lines instead of 1. We're going to see that the shape that molecules make actually affects how they work in biological systems. So that actually affects how they work. When you only have something that is 2 things, like H2 or O2 or even HCl, which is hydrochloric acid, those things make a linear shape, so they make a line kind of shape. Molecules such as methane that have 5 atoms in them make a tetrahedral shape, and so we're just going to look at what that looks like. So this is methane. This is the shape it makes when we draw out the structure of it. This just shows the electrons being shared. So again, that this is 50-50, so this would be a non-polar molecule. This is the actual shape that methane makes, so you should still see that this is CH4. This is the tetrahedral shape that they were talking about. Okay, so now we're going to go again into nonpolar and polar covalent bonds. So there's 2 types of covalent bonds. They can either be polar or they can be nonpolar. If they share the electrons equally and fairly, that is called nonpolar again. And if they don't share the electrons equally, that is called polar. We can tell whether or not they share them equally by what's called their electronegativity, which is just a small number on the periodic table, represents their ability or their muscle strength to be able to get the electrons to orbit around them. So between hydrogen, for instance, remember we talked about that it's 50-50, right? So this would be an example of a non-polar molecule. Here also the electrons are shared equally, so this is also a non-polar molecule. We're going to see in water, water is one of the ones that we use very often, but water actually doesn't share them equally. Oxygen is actually better at getting the electrons to orbit around it. So oxygen is actually better at getting the electrons to orbit around it. So instead of it being 50% of the time the electron from the hydrogen might be orbiting around the oxygen and 50% of the time it might be around the hydrogen, it's probably more like a 75-25 kind of split. So because it's not equal, we call this molecule a polar molecule. Polarity within a molecule causes the hydrogen atoms in one molecule to attract the oxygen atoms in the other molecule. So even between molecules, so even between different water molecules, they can actually be attracted to each other. So this is like one water molecule, right? But what happens is that when another water molecule comes into play, it actually lines itself up so that it's almost got like this pretend bond between it. So this is another water molecule. This hydrogen is attracted to this oxygen over here. The oxygen from it might be attracted to the hydrogen from this one. So they line themselves up so that they have this kind of pretend bond between them. This pretend bond is actually called a hydrogen bond, and it actually gives water a lot of the properties that it has. So one of them is that it boils at 100 degrees and freezes at 0. These are actually very special properties that water has that lots of other compounds actually don't have. So this is a better drawing of some water molecules so that you can see them. These dotted lines are supposed to represent the hydrogen bonding between them. But again, notice that this is the hydrogen of one, this is the oxygen of another, hydrogen of one, oxygen of another, right? And so they kind of line themselves up to be attracted to each other. So we're going to talk about the chemistry of water here shortly. The first cells evolved in water, and actually organisms like yourself are composed of 70 to 90% water. water is a polar molecule. Water molecules form hydrogen bonds, which causes them to actually cling to one another. So we're going to see that water is liquid at temperatures typically of the Earth's surface due to hydrogen bonding again, just like we had just previously talked about. So you have to know the properties of water. So this is actually the first property of water. Water has a high heat capacity. And why this is important to life is the amount of energy that it takes, so the amount of heat energy it takes to raise 1 gram of water by 1 degree Celsius is actually quite a bit. It takes about 4.19 joules of energy to raise 1 gram of water by 1 degree Celsius. This is significant. If you look at any other element on the table, any other compounds, often they are like a decimal number, so they might be like 123, like 0.123, compared to 4.19. So these ones take hardly any energy to raise the temperature of them. This one takes a lot of energy to raise the temperature of it. This is a good thing for us because we don't want to raise our whole body temperature in swift movements, right? Because that would be bad for our body. Water also, so the second property is water also has a high heat of vaporization. So converting 1 gram of the hottest water to gas actually requires 540 calories of heat energy. So to do this, it takes a lot of energy to make it into a gas also. This is actually the reason that you sweat. You're sweating to actually cool your body down. It also helps moderate the temperatures along coastlines. Thirdly, water is a solvent. So remember that we talked about water being polar, right? That oxygen is better at getting the electrons to orbit around it than the hydrogens. What this does is that there's actually a whole thing that says that likes dissolve likes. What that means is that polar molecules dissolve other polar molecules. Now, if you've ever tried to dissolve water and oil together, so tried to mix them together, you'll see that they actually don't mix together. And that is actually because oil is nonpolar and water is polar. And so they don't actually go together, they don't mix together, no matter how hard you could shake them together, because they are not the same polarity. Because water is polar, it actually makes it a really good solvent for all the things that we need to dissolve that we take in. We're going to see these words a couple times, not only here, but in other chapters where we're going to see the word hydrophilic. Hydro just stands for water, of course. Philic actually means loving in Latin, so this is a water-loving molecule. And hydrophobic, if you have a phobia of something, you have a fear, right? So this is water fearing or water disliking. So this just shows you a salt ion being dissolved by water. What you'll see is just notice the orientation of the water molecules. So this sodium ion is then attracted to the electronegativity of the oxygen. Here, the chlorine ion is actually attracted to the hydrogen molecules. Water molecules are also cohesive and adhesive. So this is the next property in water. So this is number 4. So water molecules actually cling because of hydrogen bonding. That's called cohesive. And water molecules actually have positive and negative poles that allow them to adhere to polar surfaces. That's called adhesion. So this actually makes water an excellent transport system. So inside of our capillaries, if you can imagine, it clings to the sides of the capillaries, so it helps to transport things throughout our body. Next, water has a high surface tension. So the stronger the force between the molecules in the liquid, the greater the surface tension. This actually allows insects to walk on the surface of a pond. So if you've ever seen what we call water spiders. Frozen water is less dense than liquid water. So this allows water, ice, to actually float on the top of water. This allows us to have lakes where the fish will still survive during a harsh winter, right? Because the bodies of water freeze from the top down. This is just showing us the density of water and the changes in water. This is again is the thing that allows water to be on top when lakes freeze. Next, we're going to talk about acids and bases. So acids and bases, the definition of what an acid is it's something that releases hydrogen ions, and a base is often defined as something that gains hydroxide ions or loses hydroxide ions. Here we just see water being dissociated or broken apart into hydrogen and hydroxide. So acidic solutions are always high in hydrogen concentration. So acidic solutions have lots of hydrogens in them, so hydrogen ions. Acids are actually defined as substances that give off hydrogen. So an example of this is hydrochloric acid. You actually find this one in your stomach, but you'll see that this actually dissociates to making hydrogen and chloride ions. Basic solutions are low in hydrogen concentration, so they don't have a lot of hydrogen. They're the opposite of an acid, of course. Sometimes they dissociate in water and release hydroxide ions and that take up the hydrogen ions. For instance, sodium hydroxide is an example of a very strong base. When it dissociates, it gives off sodium and hydroxide. These hydroxide really like hydrogens, and so they actually go together to make water molecules. This scale shows us the pH scale. The pH scale is just a level of measuring the hydrogen ions rather than measuring them in these weird, hard to read often numbers. The pH scale goes from 0 to 14. From 0 to about 6.99, that is an acid solution. From 7.1 all the way up to 14, that makes it a basic solution. So this is just showing you the difference in the two. So anything that has a pH, of course, below 7, so that would be this way, of course is going to be an acid. That means again that it's going to have more hydrogen than hydroxide. Anything that has a pH of 7 and above is going to be a base, so that means it has more hydroxide and less hydrogen ions. And the only time we actually call it neutral is if it's exactly the same. So if it is at 7 exactly, then the hydrogen is equal to the hydroxide. This just shows us again the pH scale, again going from 0 to 14. This shows you some of the things that you can commonly find in your household or in yourself even. So here's like your stomach acid is about here, lemon juice is about here, baking soda, which you have at home, right, is about here. The thing that you take if you have an upset stomach often or acid reflux is actually made out of baking soda. Now we're going to talk about buffers. So a buffer's job is to actually keep the pH within normal limits. So remember that your blood pH was 7.4, and your body actually likes to maintain that homeostasis of your pH being that particular number. And so it has this particular buffer inside of it, and you do need to know this buffer. So it does have these things. It's called bicarbonate ions, and this is the symbol for bicarbonate ions. And it also has carbonic acid, and this is the symbol for carbonic acid. And we're going to talk about how these work next. So these reactions here, I would strongly suggest knowing. So if, for instance, for some reason in your body, extra hydrogen ions are added to your blood, what happens here is that the hydrogen will act with the bicarbonate. Remember that these ones are the bicarbonate ions. So the bicarbonate ions will hook up with all these extra hydrogens to make carbonic acid. This eliminates all these free-floating hydrogens around in your blood, which is good because we don't want your blood to get below 7.4, right? In the next case, what happens is if, for instance, your body gets too basic, so if you have hydroxide atoms added to your blood for some reason, what happens here is that then the carbonic acid part of your buffer in your blood is going to work with the hydroxides to take up all those extra hydroxide ions, and it will actually make some water. So it'll actually make some water, and it will make some more bicarbonate ions. So these reactions actually prevent the pH from changing significantly, which is important to our body. Now we're going to look at organic molecules. Sometimes they're called macromolecules, but organic molecules always have carbon and hydrogen. For instance, for them to be carbon, they have to have carbon and hydrogen. They're actually called hydrocarbons, which is really easy, right? Because hydro looks like hydrogen and carbon looks like carbon. So they have hydrogen and carbon. This is an example of a hydrocarbon. We're going to see that the chemistry of carbon accounts for the formation of a great variety of organic molecules. Carbon actually forms 4 different bonds, and because it can form 4 bonds, it's special. Not many other things on the periodic table can actually form 4 bonds. So this makes carbon able to make lots of different long chains and also bond to lots of different things. We're going to see that functional groups are a specific combination of bonded atoms that are always reacting the same way. So the more common functional groups are listed in 2.1. So this is some of the functional groups. So things like hydroxyl is a functional group, carbonyl, carboxylic, amino. What happens is that they are just added to a long carbon chain. So these are things that are supposed to represent the carbon chain, and then these things are added to the carbon chains. So now we're going to look at macromolecules. Again, macro means large. So we're going to look at monomers. Mono means one. Monomers are like the simple organic molecules that exist individually. I like to compare them to like the individual Lego pieces, so like one Lego piece. And a polymer is what happens when I add a whole bunch of Lego pieces together. So if I add a Lego and another Lego and another Lego and another Lego and another one, that would make a polymer because I'm combining a whole bunch of monomers together. Another couple common reactions that we really need to focus on are these two. So you're going to see these reactions again and again and again and again and again, almost through every single chapter that we do. But dehydration reaction, if I'm dehydrated, think about what that means. That means I'm losing water, right? So I'm losing water, and that's exactly what it does. It actually loses water. When it loses the water, what happens is that things will stick together. So lose water and things stick together. So for instance, when I wanted to stick my Lego piece with another Lego piece, what would happen here is that one would have a hydrogen, one would have a hydroxide. Those two come off. When those two come off, that's actually how my Legos stick together. So I can show the water over here, and I can show that the Legos are now stuck together. Sometimes this dehydration reaction is actually called condensation synthesis also. The next type of reaction is the opposite. So if I wanted to break my Lego pieces apart, so if I want to go the opposite way now, what I do is I actually add the water in. So when I add the water, it breaks them apart. So this is just showing us the two different types of reactions just so that we can see. So this one again shows us that if I take water out, what will happen is that the two monomers will stick together. Here, if I wanted to break the two monomers apart, so if I wanted to break the two monomers apart, I have to add water, and then it will break up into the two individual Lego pieces again. Next, we're going to look at the different kinds of macromolecules. So this is the very first type of macromolecule is carbohydrates. Carbohydrates are great for quick fuel and short-term energy storage in organisms. They actually play a role we're going to see in woody plants, in bacteria, and even insects. They're also involved, remember that we looked at the glycolipids and the glycoproteins. Those are examples of cell-to-cell recognition. So I'm being able, again, to be able to recognize your cell from other cells. There's a couple different kinds of carbohydrates. So this is this monomer of carbohydrates. Let's just try that again. Monomer. This is the monomer of carbohydrates. It's called a monosaccharide. And notice it has one, right? The word one. Saccharide means sugar. So this is a one sugar molecule. Things like pentose and hexose. And you'll notice anytime you see the word OSE, that tells you that that's a sugar. But pentose and hexose are examples of monosaccharides. This, for instance, is glucose. These are all glucose. It's just showing you different ways that they can draw glucose. So this one and this one and this one are all exactly the same. In this first one, all they've done is shown you every single bond, every single hydrogen, every single oxygen that's attached. They've also shown you where all the carbons are at the corners here. This one, notice that the carbons are just left out. It's just assumed that you know that the carbons are there. And here, they don't even draw any of that stuff on. They just assume that you know everything. You will never be asked to reproduce this. You will never be asked to reproduce this or this. It's just important that you recognize that it's a six-sided shape and that it often represents glucose. We're not going to ask you to draw out the glucose molecule ever. The second type of second type of carbohydrate is a disaccharide. Notice again that it has the word two and a sugar, right? So this is examples of two sugar molecules. When I add a monomer of glucose and another monomer of glucose together, I get this maltose. This maltose is an example of a disaccharide. Notice here, this is called dehydration reaction, right? When I go this way. Also notice here that the hydroxide from this molecule and the hydrogen from this molecule are highlighted to show you that they come off and as a water molecule over here. When they come off, where they were before, now they stick together. So now they stick together. So we have a disaccharide and a water. Notice that if I wanted to go the other way, so sometimes in your body, for instance, at nighttime, your body needs sugar to work, and it usually needs the monosaccharides. What it will do is it'll take the disaccharide, add some water, and break it down into individual monosaccharides. So going that way is called the hydrolysis reaction. And the last type of carbohydrate is called a polysaccharide. So we're going to look at a couple different kinds of polysaccharides, and you need to know that starch, glycogen, and cellulose are examples of the three types of polysaccharides. So starch is the first one. Starch is a storage form of glucose, and it's found in plants. We're going to see that glycogen is the one that you would find in your liver. So your liver stores sugar this way. So when you eat a big piece of chocolate cake, your body can't absorb all that sugar. So what it does is it stores that sugar in your liver for later on when it doesn't have any sugar. We're going to see that this molecule here is, remember that, so you don't have to draw the glucose molecules out, but here's one glucose. Here's one glucose, right? And so when I have a whole bunch of glucose all together, notice the big long chain that it makes, and this is an example of what starch looks like. This one would be an example of what glycogen looks like. It's a little bit more complex, right? More bonding in between all the the glucose molecules there. And then cellulose. Cellulose is the one form of polysaccharide that we find in plants also. It's actually found in cell walls. Our body is not actually able to digest cellulose, so we can't digest it because of the strong nature of the cell walls found. Chitin is another found in the exoskeleton of crabs and another structural polysaccharide. So this just shows us what cellulose looks like. Next, we're going to talk about lipids. Lipids are the next type of next macromolecule. So now we're on to the second macromolecule. So this is number 2. Lipids actually have more energy per gram than any other biological molecule. So if I was going to talk about the thing that makes them unique, that would be one of them. The other thing about lipids is that they are not water soluble. So they are not water soluble. There's a couple different types of lipids, so fats and oils, phospholipids, which we're going to see in the membranes of our cells, and steroids are all examples of lipids. Lipids are, again, diverse in structure and function, so they have lots of different kinds of lipids. Lipids have one common characteristic, and that is that, again, that they don't dissolve in water. They're hydrophobic. Remember that they're scared of water. Fats are usually of animal origin. They're solid at room temperature, and they store energy, insulate against heat loss, and form a protective cushion. We're going to see things like lard is an example of a fat. Butter would be an example of an animal fat. But if you think about those things, they're solid at room temperature. Oils, those are usually from plants, right? So think about canola oil. That's a liquid at room temperature. A fat molecule is also known as a triglyceride. So it's triglyceride has the word 3 in it, but what it has is 1 glycerol and 3 fatty acids. So this shows you what a triglyceride looks like. It's a glycerol molecule with 3 fatty acids. Again, you're never going to have to draw the glycerol or the 3 fatty acids. It's just important that you know that it's made out of a glycerol and 3 fatty acids. Here, this is just showing us before it actually becomes the fat molecule. So it's the glycerol all by itself, then the fatty acids. Notice we have dehydration reaction, right? So you'll notice the hydrogens from these ones are all highlighted, the hydroxides from these are all highlighted. We're going to make 3 water molecules come off, and then it all sticks together like our big Lego piece. We're going to talk about emulsification. Emulsification is just what happens when fat actually can get dissolved. So a way that this happens is actually with soap. Soap is an emulsifier. So soap is actually an emulsifier. What it does is the fat droplets disperse in water. Emulsifiers have a polar end and a non-polar end, and the polar end actually attracts water while the non-polar end attracts the fat. This allows the emulsifying agent to wash away or carry fat away with it. So for instance, if this is my fat molecule and I wanted to dissolve it, what happens is that when I have soap, soap has this polar end. This one likes the water. And then it has this non-polar end. That doesn't like the water. So a fat molecule will allow the soap to be kind of injected into it by putting the non-polar end in and having the polar end stick out. And then what happens is that the water can actually come in and break this up because it will attach to the polar ends there. We're also going to look at trans fatty acids. So a fatty acid is a hydrocarbon chain that ends with the acidic group COOH. Saturated fatty acids have no double bonds between carbon atoms. So they're full of hydrogens. There's no double bonds, there's no triple bonds, there's only single bonds. It has all the hydrogens it can possibly hold. In unsaturated, what they have is they have 1 or more double bonds or even triple bonds between things, but they're not full of hydrogens. So this is an example of a saturated molecule. You'll see that there's no double bonds, it's full of hydrogen, it's holding as much hydrogen as possible. This is an example of an unsaturated molecule. It has double bonds in it, so you can see that it's unsaturated. Next, we're going to talk about phospholipids. So phospholipids are comprised of 2 fatty acids and a phosphate group, and we're going to look at this again in chapter 4. But they're made out of a phosphate group and then 2 fatty acids that came off of it. So this would be the phosphate group and then 1 fatty acid here and then another fatty acid. The phosphate group likes water and the fatty acids don't like water. This is just a better drawing of it just so that you can see. But again, it has the phosphate group at the top that likes water and then the fatty acid chains that don't like water. We're going to see that this actually makes the inside of our cells, the outside of our cells. So this blue part here is made with this phospholipid bilayer. It's called a bilayer because it has 2 parts to it, right? There's one part of phospholipids here and then another part of phospholipids here. Another type of lipid is steroids. So steroids, what happens is that you have those carbon rings and they're all fused together. And so that makes things like cholesterol as an example of a steroid, testosterone, and estrogen. Our next macromolecule group is proteins. So proteins are polymers composed of amino acids. Amino acids are the monomers in this case. So amino acids, often we represent them with an AA, but they are the single unit ones, the single Lego pieces. So here you'll see that these are 2 different amino acids being added together. Again, notice that the hydrogen from one and the hydroxide from another are highlighted to show that they come off as water afterwards. And then they're going to make this what we call a dipeptide. Notice that it's very similar terminology, right? A 2 sugar one was a disaccharide, a 2 protein one is called a dipeptide. So when they take off the water, that's called dehydration. When they add water to break it apart, that's called hydrolysis. So this is just showing us some different types of amino acids. There's 20 actually amino acids in total, but these are the ones just showing you the difference between them all. So you should just see that, like, basic stuff is that they have the same structure, and then to make them different amino acids, so they add different things to them. So just the part in brown. Again, never going to ask you to name the amino acids, never going to ask you to draw the amino acids, just that you recognize that amino acid is a single monomer unit of proteins. Proteins perform many functions. So they have given support, so things like keratin and collagen, which are things you find in your skin and your nails and your hair. These things give support to you and to your body. Proteins can actually act as enzymes also. Enzymes, remember, speed up chemical reactions, so they make reactions go faster. We also find proteins in hormones, which are a type of chemical messenger in our body. We're also going to look at, when we look at the muscle chapter, actin and myosin, which are proteins that help their cells move. This is actually how your muscles move. And then we're going to see that we have some proteins that actually are right inside each red blood cell that we have. So that's called a hemoglobin. That's the thing that attracts oxygen and carries it throughout your body. We're also going to see that proteins act as antibodies, so they protect our body. And again, remember, we're going to look at this one next, but they act as channels to allow substances to cross membrane. A peptide is what happens when we add two amino acids together. So remember that when we add an amino acid, a single unit, and another single unit together, we take a water off of them. They become a dipeptide, which is just a two amino acid word. So it just means that we have two of them together. So where the water was is where they bond together. It has a special name. It's called a peptide bond because it is between two amino acids. Proteins have 4 levels of structural organization. So they have what's called a primary level, which is just a single linear line. It looks like a beaded necklace or a beaded chain. The secondary one is a little bit more complex. If you ever made those fans as a kid and you folded the paper back and forth, that's an example of beta sheet, which is what that folded sheet kind of would look like. So this is just showing you the primary structure, so it's just a single band of a whole bunch of proteins all together. This is showing the 2 different types of secondary structure, so it can be that pleated sheet, or we can make this helix structure. The helix one is the one that we use in DNA. The next structure is a tertiary structure. The structure is, tertiary means 3, so it's the third dimensional structure of a protein. This is when some of these other ones get combined together. So we actually take the first and primary one and the secondary one and we combine them together. And lastly, the next one, the fourth one, it doesn't look like much, but it's actually called a quaternary structure, which means fourth. But it's when it looks like a big mass or a mess of a whole bunch of proteins together, and they even look very disorganized, but it actually does have an organization to it. This is the one, by the way, that makes up the hemoglobin that you find in your blood. So this is just showing your tertiary structure, so that you can see that it has some of those secondary structures in it, and they're now bonded together. And then when we do this even more, what we get is this quaternary structure. Again, this is the one that you find in hemoglobin. This is just showing this all. So the final structure of a protein and the shape of the protein is actually very important and tells us what it actually does. We're going to talk about what happens to proteins if they get too hot or if the pH changes too much, but they get what we call denatured. If you cook an egg is a protein, and when you cook the egg, think about what happens to the egg. It turns white. Could I get the egg back into the eggshell as that kind of gooey mess that it started as? No, right? Because what you've done is you've denatured the proteins in the egg. And so the same thing will happen in your body if your body gets too hot or the pH changes significantly. So if that buffer that we talked about before, the bicarbonate and the carbonic acid, don't work properly, then you can denature the proteins in your body, and this is really bad. You can't get them to go and work again after that. We're going to talk about nucleic acids, which is the 4th type of macromolecule. So nucleic acids are 2 types. We have DNA, which is deoxyribonucleic acid. I would make sure that you know that DNA stands for deoxyribonucleic acid, and that RNA is ribonucleic acid. We're going to see, we talk about DNA all the time on TV, right? But DNA is looks like a ladder, and then all you do is kind of twirl the ladder around. I'm not going to try and draw that because it would not be great. And then RNA is what happens is when I want to replicate my DNA, I take half of this, and then that's actually called RNA. So this is showing us that both DNA and RNA are polymers of nucleotides. Each nucleotide is actually made up of a couple things. It has a phosphate group, it has a pentose, remember that was our monomer of a carbohydrate, and it also has these nitrogen containing bases. So it has adenine, thymine, guanine, and cytosine as bases. The only thing that changes is in RNA, they have all the same ones. The only thing that changes is that you have uracil replaces thymine. So this is the 5-carbon sugar. This is the phosphate group, just so that you can see them. This just shows you a comparison between the two, again showing you the bases, right, between DNA and RNA, shows you the names of them, shows you that this one has a double strand, so like this is like my ladder example, right, and this is telling you that it's only half the ladder example. This tells you helix, so helix just means this thing twisted, and this one of course can't be a helix, right, because it only has half of the ladder. This is the structure of DNA. So the structure is that DNA forms that double helix of the 2 strands. So it has this again, and then it's just twisted. The 2 strands are actually held together by hydrogen bonds. The other important thing is to know these things. So adenine is one of those nitrogen bases. It always goes with thymine, and cytosine always goes with guanine. So even here, you can see here's adenine and thymine, they're yellow and blue, but notice here's yellow and blue, here's yellow and blue, here's yellow and blue, right? And then these two always go together, so you'll see cytosine and guanine's here, cytosine and guanine's here, so they always pair together. They don't pair with anybody else. This just shows you that again, so the pairs between the two. RNA is only half of that strand again, and so that's involved in making more DNA. The other really important molecule that we're going to talk about again and again and again is adenosine triphosphate. So I would make sure that you know what ATP stands for, that you know the words adenosine triphosphate, meaning that it has 3 phosphate groups. This is the energy or the currency of our cells. So this is the energy or the currency of our cells. When ATP is broken down, it turns into ADP, which stands for adenosine diphosphate, so now it only has 2 phosphate groups. It gives off a phosphate, and it also gives off energy. And so this is like the money of our cells, and this is how we spend the money. So we make energy for our cells to do all kinds of things. So this diagram that's kind of cut off here, but you can see that this is the adenosine triphosphate, so this is the ATP. This is where it becomes ADP, so adenosine diphosphate. Here's the extra phosphate group coming off, and then energy is given off. Again, ATP is the energy of our cells. We need it to do many, many reactions. So this is the end of chapter 2. You should go on to chapter 4 after this. Make sure that you work on the review questions and the definitions. Good luck.